Hear me? Excellent, thank you. Can you see me?
No.
All right, we can be seen. All right, my name is Manoj Dadlani. I'm the CEO of Cosmos ID. Thank you all for joining us today. Just gonna do a quick introduction, and then we'll get into the main event here. And I will share screen. Okay. Just to confirm, Rosie, Jim, can you guys say hello?
Hi, I'm here, but I don't see myself, Manoj. I only see an R, so I don't see you or Jim either on the top anymore.
All right, to the audience, can you guys see Rosie and Jim? All right, I think everyone can. Oh, nope. Looks like everyone can see us. We can't see ourselves, so let's just roll with that.
Okay.
Okay. Again, so thank you guys for joining us. As you can tell, we've got Dr. Rosa Krajmalnik Brown and Dr. James Adams. And today they'll be talking about microbiota transplant for autism. Just before we get into that, I'm just gonna give you a quick overview on Cosmos ID. We are a company that provides end-to-end metagenomic solutions. We work with pharmaceutical companies, nutritional companies, academics, and other industry partners. And we're focused on just understanding and characterizing what's in the microbiome for research and diagnostic purposes. So we work end-to-end from study design, sample collection, DNA sequencing, and bioinformatics and interpretation. We support, as I said, end-to-end, and we also support companies that are building tests and provide everything from kitting all the way through reporting. This is our current facility. We're actually really excited to be moving to a new facility. It's about 4 times larger in the next month. But here is where we provide all types of sequencing services in a regulated environment. We're CLIA and GCP compliant. Experience in many sample types and many sequencing platforms. So as I said, many sample types. We've worked with tons and tons of different types of samples. Obviously, stool samples are one of our major ones, but everything from oral, vaginal, skin samples, environmental samples, and the likes. Just a microbiology refresher. Strain is a clinically informative and actionable unit. So if you remember from your good old days in microbiology, it goes kingdom, phylum, class, order, family, genus, species, strain. Generally in microbiology, when we're identifying organisms, we're kind of playing in this region, with 16S being more focused on genus level accuracy and getting into shotgun metagenomics, where you start getting into species and strain level. As a company at Cosmos ID, we've really focused again on this strain level, and we've built databases and algorithms to be able to interrogate sequence data to get accurate strain level resolution. And we've kind of proven that through multiple third-party validations. We won Janssen Pharmaceuticals Mosaic Strain Challenge with the best strain level accuracy for microbiome analysis. And we also won both of the Precision FDA challenges in microbiome analysis. And again, this is based on our bioinformatics and the curated databases that we've built and the algorithms that we've built to mine sequence data. And then what we've done as a company, we obviously do the sequencing side of it, but the main value add really comes from the bioinformatics and how we're able to give very accurate microbiome analysis. So multi-kingdom, bacteria, virus, fungi, protists, functional characterization such as antimicrobial resistance genes, virulence factors, ammonia-producing genes, things like that. but then we also package it in a very easy, user-friendly way to interrogate that data. We work in many industries, so as I mentioned before, pharma, nutrition, infectious disease diagnostics, animal health, cosmetics and personal care, and academia. And these are some of our amazing partners that we've been working with over the years, and we're very thankful for the partnership and the businesses that we've worked on together. And with that, from our autism webinar series, I really want to thank our sponsors. On one of the previous webinars, you heard from Microbiome Foundation, and today you'll hear from Nirvana Biome. So Kelly, I'll hand it to you.
Thanks, Manoj. Hi, everyone. Thanks for joining us today. I'm Kelly Moffitt, Director of Microbiome Health at Nirvana Biome. And next slide. Manoj, next slide, please.
Kelly, can you hear us? I can't hear you.
Oh, I can hear you. Oh.
All right, Kelly. Everyone else can hear you. Please go on.
All right, sorry about that. We're having a few hiccups with some of the people on here being able to hear and some not. But moving on, I just wanted to make a few notes that we have an autism research program at Nirvana Biome. We use whole genome shotgun sequencing to provide really great taxonomic and functional identifications and information. And if you are interested in this program, please email us for more details, but we provide a report as we learn more about the microbiome and autism spectrum disorder. We will send updates, and we also can compare microbiome samples before and after fecal microbiota transplant. Next slide, but he can't hear me. And this is an example of some of our reports. So we get kind of an overview of what's going on in the microbiome as well as autism-specific information. I'm just chatting and typing into the chat. Next slide. And just a little sample of some of the research we're doing. This is a component analysis of children with ASD, children who do not have ASD, and adults who do not have ASD. And you can just see from this pretty small sample size that we see these distinct clusters. And this is kind of guiding our research in this field, knowing how different and distinct these microbiomes are. And for this one, just a few of the different genera that we know are playing a role or at least affected in these different types of microbiomes between children with autism spectrum disorder and children who do not have it. And so, this is just the beginning of our exploration, but as we learn more, we share more with our clients. So I think it's really an exciting time here. And I think that, if there's anything anyone has questions about, please just follow up after. And lastly, we have a discount code for our autism spectrum disorder kits. So it is good until June 15th, and it's 15% off, and the code is ASD15. All right, so thanks for bearing with us through these technical challenges, and I'm just sending it back to Manoj.
All right, thank you, Kelly. So let's do some introductions of our speakers. We'll start off with Dr. Rosa Krejmalnik-Brown. She's a pioneer in research on gut microbiome and autism, specializes on molecular microbiology, microbial ecology in two main functions, one for bioremediation and the other in human intestinal microbiology. Again, with relationship to autism, as well as obesity, bariatric surgery, and metabolism. She's the director of the Biodesign Center for Health through Microbiomes at Arizona State University, where she's also a professor of the School of Sustainable Engineering and the Built Environment. She received her PhD in environmental engineering from Georgia Tech. She recently won the great honor of Arizona Researcher of the Year by AZ Bio, and has some other amazing awards in the past. And she also has 4 patents and over 115 peer-reviewed publications. Dr. James Adams, he's the director of the Autism Asperger's Research Program at Arizona State University. His research focuses on the medical causes of autism and how to treat and prevent it. And some of the major areas that he's focused on are nutrition, oxidative stress, gut problems, and then gut bacteria, toxic metals, and seizures. He has over 150 peer-reviewed scientific articles, including 50 that relate to autism. Some of his prestigious positions are president of the Autism Society of Greater Phoenix, president of the Autism Nutrition Research Center. He's the co-leader of the Scientific Advisory Committee on Autism at the Autism Research Institute, and he's also the chair of the Scientific Advisory Board of Neurological Health Foundation. And he has an adult daughter with autism. And with that, I will hand it over to Rosie and Jim to walk us through some of their amazing work.
Jim, can you hear me? And can you share your screen? Manoj, I don't see Jim or the slides. Okay, slides are coming. Good morning for us. Good afternoon in the other side of the country. Thank you for inviting us to give our talk and present our research on microbiota transplant therapy for autism. Next slide. Before I start our presentation, I want to make sure that we acknowledge a lot of people that participated in this research. Dr. Daewoo Kang, who is here in the middle, in the top, was a postdoc in my lab, now is an assistant professor, and he led most of the research that we're going to be presenting. Dr. Kemlar Nirmalkar is a postdoc in my lab. Next to him, under that is my research team, but we also had a long list of collaborators. On the left of this slide, you can see them, and lots of families who participated, donors, etc. We also have a few funding agencies that we want to acknowledge: Arizona Board of Regents, ARI, ZUOC for Autism, and many more, but we want to make sure we don't forget this before we start the presentation. Next slide. Jim and I have pending patents for FMT and autism. We both have worked with Prestovo and Finch Therapeutics and have provided some consulting for them. I want to remind everyone that MTT, which is how we call our modified FMT, is an investigational treatment and is not approved by the FDA for autism. And Jim and I are co-owners and initiators of a startup company, Autism Diagnostics. These are our conflicts of interest. Next slide. When I started doing work on the microbiome, there were 2 pieces of evidence that convinced me that asking the question on if there's a possible contribution of the microbiome to autism was a question worth investigating. And this is one which is research done by my collaborator, Jim Adams, where he showed that there is a strong correlation between gastrointestinal symptoms and autism severity. In this figure, you can see on the x-axis, autism treatment evaluation checklist, which as the number is higher, the severity of the disorder is stronger. And on the y-axis, you can see gastrointestinal score index, which it's the same as the index is higher, the severity is stronger. You can see that children that have strong GI symptoms also were more severe in the scale of autism. Next slide. Also, seminal work from the group of Sidney Feingold in 2000 showed that they had a group of children with autism that were treated with vancomycin. And in this slide, what you can see is improvements during treatment. Most of the children improved while they were getting the vancomycin treatment, which is an antibiotic that it's not absorbed, so it makes it to the gut. And the interesting thing of this study was that when the children stopped taking vancomycin and there was a follow-up visit, many of the improvements were lost. But this showed a strong interaction with the microbiome. Next slide. The microbiome is unique to each individual, and we know that there's general stability through life, and I have general in quotations because today we're going to show you that we were able to change the microbiomes of some of these children. It's affected by internal and external factors, and these include the environment. This can be the external environment or the host environment. It's altered in disease, and it's strongly affected by residence time, which is something that it's really important, especially for our research, where we know that constipation and diarrhea will affect the residence time. And we also know that we have about 100 times more genes in our body from microbes than genes from our own genome. Next slide. So as I said before, at the gene level, we're 1% human and 99% microbe. And this has awakened a lot of research in the area of microbiome. And on the left side of this slide, you can see a lot of disorders or diseases that have been targeted to the microbiome. In red, I have obesity and metabolism and autism, which are two topics that my research team is addressing. But I also want to mention that there's a huge opportunity for impact here, because if we can modify the microbiome to a beneficial microbiome, the impact could be better metabolism, better GI health, better behavior, better mood, less obesity, and reduced type 2 diabetes. And we could also modify drug activity by modifying the microbiome. So as engineers, we always look at not the problems, but how can we solve problems? And we think that there's a lot of potential and opportunity here by modifying the microbiome. Next slide. The gut is where most of these microbes that I just mentioned reside. A huge percentage of the microbes that we live in our body live in the gut. And these microbes have many functions, starting from very simple ones like breaking down the food that we eat into smaller chemicals or some smaller substrates, but they also produce vitamins for us. They produce short-chain fatty acids, which have been implicated as signals for health and potentially with the brain also. And they produce metabolites, which some of them could be neuroactive and could be interacting with the brain directly or through the gut and also with the liver. Next slide. There has been a lot of research done on the microbiome and autism. There's maybe more than 20 papers on what's the difference in the microbiota between typically developing individuals and individuals with autism. And I know there's many, so I just want to summarize a few key findings from many of this. Some of them look at increase of harmful microbes like Sutterella and Clostridium, but others like us look at lack of beneficial microbes. And that's what we have found in our previous research, which is our focus right now. And so we found Bifidobacteria, Prevotella, Lactobacillus being important, and we also found lower diversity in most of our studies in our children with autism. Like I said, many studies, different findings. I'm going to show you a couple of slides with some of our most important findings. Next slide. As I mentioned, we found in the very first study that we did that when we compared children with autism with typically developing children, by using two different estimates of diversity, you can see on blue or typically developing or neurotypical children, and on red or children in the spectrum, ASD. And you can see by these two different metrics, we always found that the typically developing children had higher diversity. Next slide. we look at some specific microbes and some specific fermenters that were missing in these children, and one that we have been following is Prevotella. And here on the right, you can see on blue on the top, it's our neurotypically or typically developing children. This is a heat map that shows relative abundances of Prevotella. And on the left, you can see the red samples, which are our ASD samples. And you can see very clearly here the depletion. I want to mention that we repeated this in a second cohort in Arizona, and this was reproducible. But we recently did also another study with Kathleen Luzopon in Colorado, where she used the exact same methods for DNA extraction and sequencing to see if this could be a biased sequencing, and we could not repeat this signature of depleted Prevotella in Colorado cohort. So that's important to mention. So we saw in short that the diversity was lower and that we were missing some beneficial microbes. So, and an easy transition to come up with a solution is to say we're missing microbes, so these microbes should be added somehow. So I pass it to Jim so he can explain to you a little bit more of how we went about adding these microbes.
Give me just a moment as I restart the sharing. Okay, so as Rosie was discussing, children with autism have lower diversity, and a typical person may have of order 1,000 species. Kids with autism are missing several hundred. And so one type of treatment that's been used in the past for treating infections, a very nasty infection called Clostridium difficile, is treatment with FMT. These nasty C. diff infections cause life-threatening diarrhea in of order 500,000 Americans each year, and they kill of order 29,000 Americans a year. So very, very nasty diarrhea. It can be treated with antibiotics, but it will often return after antibiotics, and people just get weaker and weaker and can die from it. It's been shown that FMT, just taking a raw stool from a healthy person and putting it into a sick person, either by colonoscopy or by rectal enema or through a nasal gastric tube, is miraculous. It will generally cure C. diff infections in 2 to 3 days with just 1 to 2 doses, with about 90% success. And unlike with antibiotics, it essentially never returns. So FMT is now used by hundreds of hospitals in the US. They've treated over 50,000 people with C. diff infections, but it is not yet approved by the FDA. The FDA has classified human stool as a drug, the same way human blood is classified as a drug. So therefore, phase 2 trials are underway right now by our collaborator Finch and by other companies to try to get FMT approved by the FDA. Our collaborator in Australia, Tom Barodi, used FMT to treat 9 children with autism. Unlike C. diff, which only takes 1 to 2 doses, he found that children with autism were very, very difficult to treat. Their microbiome was very hard to change. So he treated them every day for 3 months and slowly, gradually saw improvements in GI symptoms, and surprisingly, to his surprise, also saw improvements in autism symptoms. So we worked with him on developing our Microbiota transfer protocol, which involves 2 weeks of oral vancomycin to kill harmful bacteria, like certain species of Clostridia, and then 1 day of fasting with a bowel cleanse to really remove as much of the contents of the bowel as possible. And then we did high-dose microbiota for 1 to 2 days, and then 7 to 8 weeks of daily low dosing, again giving that with Prilosec, which is a stomach acid suppressant. because the maintenance dose was given orally, and normally your stomach acid would kill off most of the bacteria you take orally. We were using a liquid formulation developed by our collaborators at University of Minnesota. It's a highly purified form. So starting with raw stool, they purify it so it's 99% bacteria from healthy, very carefully screened donors. They figured out how to freeze dry it so it's very stable and can be saved for over a year. And so essentially it's like giving a super probiotic with 1,000+ species of bacteria from very healthy, very carefully screened donors, generally the top 10% healthiest of the US population. So what we observed is that there was a great improvement in GI symptoms. That on the scale on the left here, we see that with 1 being no symptoms at all, and going up to a severity scale of 7, the children with autism started out with a variety of GI problems, primarily constipation, diarrhea, abdominal pain. And at the end of 10 weeks of treatment, those symptoms have been reduced by 80%. 16 of 18 children had tremendous improvement in their symptoms. 8 weeks after treatment, we held our breath to see what would happen, because with vancomycin alone, the benefits were lost within a few weeks. But by vancomycin followed with fecal transplant, those benefits held in all of the children. So now 17 of 18 children had great improvements in their GI symptoms. And then we stopped, we published our results. We thought that was very exciting. But then several parents came up to me a year later and said, Professor Adams, my son's doing better than ever. So therefore, we did a 2-year follow-up study, 2 years after treatment had stopped. It's very unusual to do a 2-year follow-up study. And thankfully, every family who participated in the study chose to respond. And so what's very interesting is that we see the children who had, 2 years later, most of them had continued to have very good GI symptoms. There were a few children who lost some of those GI benefits. In one case, it was due to taking an oral antibiotic. But in general, most of the GI benefits held. We also see one child here who was a slow responder and did eventually improve greatly. If we look at specific individual symptoms, we see abdominal pain greatly decreased. Reflux was not really present in the children at the start and still not present at the end. But symptoms of diarrhea greatly reduced and symptoms of constipation greatly reduced. So if your stool was too hard or too soft, it ended up becoming just right. We also looked at autism severity symptoms. So using a scale called the CARS, which is done by our expert evaluator. At the start of the study, most of the children were in the severe range up here, only a few in the mild to moderate range. After treatment, most of the children improved. 8 weeks after treatment, those benefits held. There was no loss, which again, with Vanco alone, the symptoms would have returned to baseline. But then, really exciting, 2 years later, only a few children remained in the severe range. Many more of the children in the mild to moderate range, and some were well below the cutoff for autism. Now again, this is an open label study without any placebo control, so there's some placebo effect, but the CARS generally is fairly resistant to placebo effect. And we have an expert, very experienced evaluator, and she said she'd never seen such improvement from any of the studies she's been involved with compared to the improvement from MTT. If we look at other scales, if we look at the aberrant behavior checklist, we saw significant improvements in irritability, in lethargy or social awareness, improvements in stereotypy or stimming, improvements in hyperactivity, and improvements in inappropriate speech. On the social responsiveness scale, we saw improvements in social awareness, improvements in social cognition, improvements in social communication, improvements in social motivation, and improvements in social mannerisms. Just all across the board, improvements in some very important symptoms and associated symptoms of autism. So now let me turn it back to Rosie.
Now I'm going to tell you what happened up to the microbiome. As you probably remember, I mentioned that one of our goals was to increase microbial diversity. And in this graph, I'm showing you that microbial diversity increased. Here we're showing faith PD, which is a metric for diversity. And you can see in this graph, on the green line are typically developing children that are controls age matched. On the orange darker line is our initial high dose donors, and on the light line is our low maintenance donor or low dose donor. And you can see that in the beginning of the treatment or at baseline, or the faith diversity of these children was very low. At 3 weeks, we didn't see a lot of increase, but these children had just been treated with vancomycin and added a dose of the high dose donor. And then you can see these increases after week 10, after week 18. And amazingly enough, you saw that the improvements were huge at 2 years, and the diversity here is much larger at 2 years, which is our follow-up. And it's much larger than the neurotypical kids. This increase of diversity, I will show you later, that is a new diversity and not just the microorganisms that we added with the treatment. My assumption here is that we managed to change the environment in the gut, and this led to beneficial microbes being able to attach and grow and thrive in this new environment. Next slide. And as I mentioned before, we looked at how much of the donor and graft, or how similar was the microbiota of the children that we treated to the microbiota of the most recent donor. And so here you can see a unweighted unifrac analysis, which is a measurement of dissimilarity. The lower the number, the more similar things are to what we are comparing. So here in the beginning, we just have how similar the children are to the donor that was being provided to them. So this, I would say, it's the average of your similarity to the person that would be sitting next to you if we were at a conference. And here you can have in the green line the average similarity between the typically developing children. And as the treatment progressed, we can see that these samples became more similar to the donor. So that's a sign of engraftment. And they were also more similar to the donor than you would be similar to another person. And that continued after we finished the treatment at week 10. We can see that some of these got lost a little bit after 8 weeks on our first follow-up, but they were still much more similar to the donor than to another child. But we can see that at the 2-year follow-up, this microbiota is not more similar to the donor than it would be to another child of the same age anymore. So this is what leads me to believe that these children had a new environment that allowed them to recruit and retain other beneficial microbes, not just the ones that we added with the treatment. Next slide. We look at some specific microbes that we've been following. Bifidobacterium is a probiotic, a beneficial microbe, and you can see in this slide, at week 0, it was much lower in these children than in the typically developing children. Again, on green on this slide, it increased just by adding the vancomycin, and by the initial dose at week 3, increased more at week 10. And we can see a slight increase week 18 and at 2-year follow-up. Prevotella, which is a microbe that I mentioned that was depleted in our first cohort, we can see that it was depleted for most of the participants in the beginning, although there's 3 that had high Prevotella. It increased substantially after 3 and 10 weeks. 10 weeks, it's a very, very large increase. And although it decreased at our 2 follow-ups, it's still much higher than in the beginning and much higher than our typically developing children, also labeled here in green. The sulfobibrio is a microbe that I will call controversial because it could be pathogenic or it could be beneficial, but we have our reasons to follow it. And it's also a microbe that was low in most of the children in the beginning and increased with treatment. Next slide. And so we showed you that we changed the microbiome, we changed probably the gut environment, and gastrointestinal symptoms improved dramatically and behavior improved also. And so one of the things that my research team is investigating is, but how? How did this happen? What's the connection? So this is a famous slide from my Nature review that many researchers show that shows many of the possible mechanisms by which the gut microbiota can communicate with the brain. And I'm just going to mention the mechanism that my research team in collaboration with Jim is pursuing, which is the production of metabolites that are neuroactive. And here I have the production, but it could be the production or consumption, because these microbes can produce or consume and change the levels of these important and critical neuroactive metabolites. Next slide. And so on the treatment that we showed, I'm not sure if Jim mentioned this because I couldn't hear him, but we took fecal samples and we also took plasma samples for analysis of metabolites, among other things. So I already showed you some of the results with the microbiota with the fecal samples, but with the plasma samples, the exciting thing is that we were able to see changes that were systemic. We could see them in the plasma. And what I'm showing you here is a PCA analysis where each dot or square is the overall metabolome of the plasma in an individual. In squares, we have our baseline for the typically development, which is our comparison. You can see that mainly on the right. On red circles, we have our ASD samples at baseline. On blue circles, we have ASD at week 3, which in this case is just after vancomycin, before we even added the microbiota. And so some of them are still here, some of them migrated already to the right. And on green, you can see our ASD samples are week 10. And one of the things that I wanted to point to your attention is that the samples at week 10 are much closely to the typically developing children. So this metabolome changed, and it looks like it changed to be towards more similar to the typically developing children. Next slide. And so we had some metabolites. This is a heat map that shows just a big picture, big idea, how we had some metabolites that in the beginning were higher in our ASD children, and then after treatment, they became lower, and some that the opposite were lower, and at the end of the treatment, they became at higher relative abundance, and they're more similar to the typically developing children. When we look at fecal metabolites, I'm showing you a heat map here. You can see that it was much harder to see a difference. This sample is a lot more heterogeneous, and it was, we really had to go with the microscope for an analogy to see differences in the fecal samples. Next slide. One of the metabolites that we have been following that we saw a difference with in the fecal samples is paracresol sulfate. Paracresol sulfate is a product of tyrosine metabolism. Tyrosine is an amino acid, and when microorganisms break down these amino acids, they can produce paracresol and a few of these other intermediates, like for ethylphenol. And I point to this figure, which is from one of the publications from the group of Sarkis Masmanian, because one of the things that they observed in their MIA mouse model was that there was a higher concentration of 4-EPS, which is this metabolite, for ethylphenyl sulfate, which is also a product of tyrosine metabolism. And that when these mice were treated with Bacteroides fragilis, the concentration of 4-EPS got reduced and the behavior improved also. In theory, the microorganisms produce the ethylphenol and paracresol, and the host makes the final sulfation step. Next slide. And so I mentioned to you before that we were interested in the sulfovibrio. The sulfovibrio is a very versatile microbe, but one of the things that they can do is sulfate production. So it would make sense, even just by its own metabolism, that it might be linked to paracresol sulfate. And here we showed that there are correlations between the sulfovibrio and paracresol sulfate. Paracresol sulfate, I didn't mention on my previous slide, did change with treatment, which is one of the reasons why we're interested. And we also saw that there's a correlation between Paracresol sulfate and sulfate, and also sulfate and the sulfonylurea, which is a metabolic, hopefully a metabolic correlation. Next slide. When we look at the metabolites in fecal samples and in blood samples, we saw a lot of changes. So I just want to summarize some of this. We think that changes that happen in the gut, which happen through our fecal microbiota transplant, you can see it on the bottom, led to different metabolites which migrate to the host through the plasma. Some of the changes that we observed in the fecal samples are related to tyrosine metabolism, paracresol sulfate, and I know that Jim is going to mention also another product of tyrosine metabolism in the next slides. But we also saw important changes of metabolites that either increased or their relative abundance went down after treatment. And in this slide, you can see when you see a blue arrow, these are metabolites that were lower and increased during treatment, and the red arrows are metabolites that were higher in the beginning and decreased with treatment. And the big categories, which are all important categories that we saw, were amino acid metabolism. I mentioned tyrosine also. Cofactors and vitamins, which I mentioned, these are some of the things that microbes do for us that are really important. Lipid metabolism, purine metabolism, which this also can be linked to oxidative stress, and xenobiotics and carbohydrate metabolism. And I know that Jim has some slides prepared to talk to you a little bit more about other sources of analysis that we did to understand better some of the changes that we saw in the metabolites and plasma. And with this, I'll pass it to you, Jim.
Thanks, Rosie. So Rosie did a great job explaining changes in the individual metabolites. We also worked with a group led by Jürgen Hahn, where we looked at multivariate analysis, looking at multiple metabolites together. And so of the 621 metabolites that were measured, 11 were significantly different in the autism group with GI symptoms compared to the typically developing children who did not have GI symptoms. So if we plot the children with autism in red and the children, the typically developing children in blue, if we plot them against these three metabolites, which were especially differentiating, there was essentially no overlap between these two groups. So the curve on the right just shows that each one of the metabolites is pretty good at classifying autism versus controls with 71 to 87% accuracy. But if we put all of them together, we then get 100% accuracy in distinguishing autism versus controls. There's no overlap. And so another way of showing this is we created a metabolite model based on 5 metabolites to create an autism scale. And so on the left is the autism group, on the right is the control group, and this is a numerical number based on these 5 metabolites. And we see that the autism group is this red curve before treatment, and the neurotypical group is this blue curve, and we see this almost no overlap between the two groups before treatment starts. After treatment, this red curve is shifted greatly to the right to become this yellow curve. So after just the vancomycin, there's a major improvement in these metabolites. They now begin looking much more like typical children. And then by week 10, on this bottom graph, shown in purple, now the autism group largely overlaps with the non-autism group in terms of these 5 key metabolites. So very compelling data that not just MTT doesn't just improve autism symptoms and GI symptoms, but it's improving the chemistry in the blood itself. And when we look at these combinations of metabolites, we find that each one of these 3 metabolites is highly correlated with 10 or more other metabolites, and they don't correlate with one another. So what we seem to find is 3 distinct areas of metabolic disruption. And some children have more of one or the other. When we look at those 3 areas, if we look at sarcosine, we see that initially most of the children have low levels of sarcosine compared to the typical children on the far right. After vancomycin, most of the kids have normal sarcosine, and at the end of MTT, they pretty much all have normal sarcosine, similar to that of the typical kids. For tyraminosulfate, there was not much change. The children started with low levels and generally ended with low levels, whereas the typical kids had higher levels. The limitation is this was right at the detection limit, so 3/4 of the autism samples, about half the control samples, were below the ability of the lab to detect. If we look at inosine 5-monophosphate, most of the children started with low levels. After vanco, it improved. After MTT, it remained improved, very similar to that of the typical children. So 2 of these 3 key metabolites improved after MTT. So not all. The one that didn't improve is a good clue for future work, but it shows there are clearly major changes in metabolism. If we look at what's important about these 3, IMP is involved in NAD-dependent oxidation, which converts IMP and NAD to NADH and xanthosine monophosphate, which is a key precursor to purine metabolism. It's also been suggested to be related to irritable bowel syndrome. IMP is significantly positively correlated with 3-phosphoglycerate, which is linked to energy metabolism in mitochondria and to folate metabolism. And we know from our previous work that that's a very important area of autism. So it's a clue that we're seeing major improvements there. Sarcosine is also known as N-methylglycine. It's converted to glycine, which has many, many roles in the body, including synthesis of glutathione, creatine, purine, serine, nucleic acids, and just general homeostasis of your central nervous system. And sarcosine supplementation has been suggested to be beneficial for depression and schizophrenia. So again, seeing improvements in that, seeing increases due to MTT is very compelling data. And then finally, the metabolite that did not improve significantly, tyraminosulfate. Those levels are much lower in autism, and 78% of the group had levels below the detection limit. It's a derivative of tyrosine. Rosie talked earlier about how important that is. It has a similar chemical structure to 2 other tyrosine derivatives, 4-EPS and p-cresyl sulfate, that are linked to autism severity. However, whereas tyraminosulfate is lower in autism, EPS and p-cresyl sulfate are higher. So it seems that when tyrosine is, it's better for it to be converted to tyraminosulfate than to go down the path of 4-EPS and p-cresyl sulfate, which are associated with more severe autism. So just to summarize, many metabolites are significantly different in the autism group, which has GI symptoms, compared to the typical developing children who don't have GI symptoms. So you don't know if the differences are due to the autism or due to the GI symptoms or due to both. We're trying to answer that in our current studies. But we do see major changes in blood metabolites after FMT. Combinations of 3 to 5 metabolites have yielded very high sensitivity and specificity, allowing us to develop a pilot model for autism that may be of order 95% accurate. We found 3 general areas of metabolic disruption. Even though many metabolites are different, it seemed to focus in 3 areas. And MTT seemed to improve 2 of those 3 key areas. So not all, but as the song says, 2 out of 3 ain't bad. More metabolic results are currently being analyzed, and we'll continue to report. We published 3 different papers in this area now. So in terms of current work, Rosie is now working on whole gene metagenomics and links with metabolites. This is funded by Finch Therapeutics, which we appreciate. We're doing an adult study funded by Department of Defense in collaboration with Rich Fry and now Stephen Walker. This is a randomized, double-blind, placebo-controlled study. We've treated about 38. Actually, it's up to a little higher than that now, and we have another 40-ish to enroll. And we've started a new child study, which is a randomized, double-blind, placebo-controlled study funded purely by children, parents of children with autism and their friends and supporters. So we deeply thank the families who donated $1 million for us to do that study, and that's underway. We've begun treating our first group. We're about to start the second group, and we're planning the third group. So that's moving along. COVID slowed us down a fair bit, but now we are back in business. The goal of these studies is to prove efficacy, to try to improve dosing and duration. It's important to realize our first study was just our first guess as to what dose to give and how long to give. And because there were no adverse effects to the microbiota itself, and it seemed to have good benefit for most children, why not try higher dosing? Why not try longer dosing? So that's what we're doing in our current studies. Maybe we can have even more improvement. We aren't sure if vancomycin pretreatment is needed. So in our current studies, half the children will receive it and half won't. Vancomycin clearly kills off harmful bacteria, but it also kills off some beneficial bacteria. So maybe it's better not to give it, or maybe it doesn't matter. The FDA has asked us to investigate that. And then finally, Rosie is leading the charge in figuring out which bacteria are abnormal in autism and trying to help us figure out how to improve that and how those abnormal bacteria relate to abnormal metabolism. And then our biomarker work, putting that all together is just so important to really try to understand the mechanisms by which these bacteria are affecting autism and GI symptoms. So with that, I want to thank everyone. Again, it's clear that the microbiome has changed in the long term from microbiota transplant to a new microbiome that's generally stable in most but not all participants. GI improvements, initially 80% reduction in symptoms, 2 years later, still 59% reduction. And partly in one case due to antibiotic usage right at the end of treatment. So we need to be cautious about antibiotics. Autism symptoms, just family after family reported, in most cases, slow, steady improvement over the next 2 years. So it wasn't instantly learning language, just seemed to be an increased ability to learn language, social interaction and behavior. So initially a 24% reduction in autism symptoms. At the end of 2 years, 47% reduction. Again, there's some placebo effect. But we hope our randomized studies will lead to us better understanding and proving the efficacy, and we hope to eventually get it approved by the FDA to help the millions of people with autism who need it. So with that, we'll open it up for questions, and you have our emails here too in case we can't answer all the questions. And Rosie, I believe you're still looking for a microbiome postdoc. And so you can contact her about that.
All right, great. That was amazing. Thank you, Rosie. Thank you, Jim. And we have a bunch of great questions that are coming through the chat. I'm going to post them in the chat because I think there's a little bit of internal communication sound issues on our side. But that way, Rosie and Jim can both see the questions as we ask them. So I'll start off. If you have more questions, please post them in the chat, and I will kick off right now. So first question is, where can we get the research paper? We will we'll follow up with everyone that attended with a link to it.
There's two research papers. Actually, there's 4. There's 2 that are on the one on the FMT, one on the follow-up, and we have actually 3 on the metabolites.
And there's also
so
one.
Manoj, if you don't have the links, we can send them to you.
Perfect. No, thank you, Rosie. We'll get the links from you, and then we'll send them out to everybody. And next question. This is from Cheryl. Are both factors, which are including harmful microbes and beneficial microbes, simultaneously found in ASD children? So is the ASD signature indicated not just missing microbes, but also an increase in harmful microbes?
Is
Rosie?
Jim saying something or should I say something?
Go ahead, Rosie.
Rosie, why don't you go? I can see Jim on my other screen.
Yeah, so I'll start and Jim can follow up. What I have seen is that some studies pay more attention to some than to others. Obviously, there's always a cooperation competition situation in the gut. So for example, when you add antibiotic and you deplete the non-beneficial microbes, there's an opportunity for microbes to grow because there's more nutrients and there's more space. So it's just a question of what environment you created and which microbes will thrive. So Clostridia, for example, has been one, I would call it family that has captured a lot of attention. And I guess part of the challenges is that it's captured attention as family, but you have beneficial microbes and pathogenic microbes in that family. So we did not see any signature, for example, with Clostridia, but I know that other people have. And there was a specific Clostridia isolated from some of the seed fungal studies, Volti, which has been associated specifically to autism. And I know Jim can add more to this, but yes, there's both situations, and sometimes you see both because once you deplete the harmful microbes, ideally the beneficial will take over, which is what you want. But both situations are not always observed.
Got it.
Yeah,
Jim, anything to add?
Yeah, I think as Rosie put it very nicely, the point is the Vanco will kill off harmful bacteria, but if you do nothing, they'll just regrow. So it seems that by replanting with beneficial bacteria, then you're able to prevent the regrowth of the harmful bacteria. So that seems to be very important. But also the beneficial bacteria play many important roles, producing vitamins, helping digest your food. So it's not just the toxins produced by harmful bacteria, but it's also a lack of all the good things produced by beneficial bacteria. A simple example is butyrate, which when you eat fiber, and most Americans don't eat nearly enough fiber, butyrate is converted by a few beneficial bacteria. Fiber is converted by a few beneficial bacteria to butyrate, which is the main food, the main nutrient for the cells that line the colon. They get 60 to 70% of their energy from butyrate. So if you're not eating enough fiber or you lack those beneficial bacteria, then you're going to not be producing enough butyrate. And so the cells that line your colon will be starving, and you should expect GI disease. So next question.
Hey, Jim. Next question. This is from Joel. He says, amazing work. Any clue as to what could have been the proper placebo or control in the 2017 trial?
I'm sorry, could you say that again?
Jim, you want to take that?
Yeah, I'm sorry, I didn't hear it. Can you say it again, Manoj?
Proper placebo or control in the 2017 trial?
So we had a control group of typically developing children who were not treated, but the autism group was treated open label. Every one of them knew they were receiving the real treatment. So now in our new studies are randomized, double-blind, placebo-controlled studies where children either receive the real treatment, the Vanco, the bowel cleanse, and the Microbiota capsules, or they receive placebo Vanco and placebo Microbiota capsules. But we have to give them the real bowel cleanse because otherwise you know which group you're in. If you have, total emptying of your gut from a bowel cleanse or not, it's a dead giveaway.
Thank you, Jim. Next question. This is from Archana. How do the results of this study compare with changes in microbiota following dietary changes? For instance, a high fiber diet. Many ASD children are known to prefer high refined carbohydrates, low fiber diets.
So I'm going to take a stab and then pass it to Jim. I hope that's okay. So my understanding, Jim has done a few interventions with vitamins, not with diets, and have not been, the results have not been as strong as what we achieve here. High fiber would be the one thing that I would always recommend, especially because Prevotella is associated with a high fiber diet. But I guess one of the challenges also is getting the children to eat these other foods. So we monitored their diet in the beginning of the study, and we asked questions as if did anything change, and we got general answers. But one thing we didn't do is to take diet diaries at 18 weeks and at 2 years, which I wish we had done, because although we got answers from the parents that things didn't change dramatically, I would hypothesize that after the children were more comfortable, had less stomach pain, they were probably more eager to explore and eat other foods that they were not eating before, and that might have also contributed to the change in diversity. So in my opinion, the combination is the best approach if you can achieve it. But you have to change a little bit of their symptoms and their mindset before you can change the diet. And I'll let Jim add if he wants to.
Yeah, I'm sorry, I do need to leave now. I thought we were finishing at this time, but Rosie, if she can stay and take any more questions, that's great. Thank you very much.
I've got two more questions if you can make the time here. This one I think you'll find interesting. It's is there information on whether fungi or fungi together with bacteria can help autism patients in some way?
That is a great question, and we actually have done a couple of small studies with fungi, but we haven't published them. I'm sorry that Jim had to leave, but Jim educated me that there was a survey from ARI where they asked the parents what are some of the treatments, this is before we did our study, that have enhanced their kids' lifestyle the most, and some of those are antifungal treatments. So fungi definitely can have an effect. So right now we are doing the analysis of fungi before and after our treatment on the treatments that we're running right now. So we did some deep sequencing for fungi for some of our previous cohorts, and we also have the way to do quantitative PCR. And so we're targeting general fungi and candida to look at this. So we're looking into, it's a great question, and it's two things. It's just the fungi, but it's also the interaction of fungi and bacteria. And the other question comes with the competition that I mentioned before, which is when you add an antibiotic, maybe the bacteria go down, but the fungi could take over because now there's more nutrients. So it's definitely an important and great question. And so Manoj, I know there might be another question, but I too gotta go if that's okay.
No, absolutely. I think the last one, and maybe you can just answer quickly, is there's multiple questions on how people can enroll in future studies with you.
Yeah, so we can send you a link, and I can tell you that the MTT right now it's oversold and full, but there's a waiting list. But we're running a probiotic study where anyone who can actually participate. So if you can distribute this with the people that attended the webinar, we can send you links.
Absolutely will. Rosie, thank you again so much, and really appreciate the amazing talk. And thank you to our audience. This will be available on demand, and we'll follow up with an email with a link to the publications and how to enroll in the study. So thank you again, and have a wonderful day.
Thanks for the invitation, Manoj.
Thank you, Rosie.
Okay,
absolutely. Bye.